Hey guys, what's going on? With Caveman Q here again, Pale Up on Long Island with caveman.com. Another edition of Monday Nights with Mike, live stream Q&A. You know the deal, ask me anything, health related. Sorry for the delay. Unfortunately, oh, hello. Sorry for the delay. Unfortunately, my Wi-Fi wouldn't let me change to the correct one. So, yeah. That being said, we are here. We are ready to rock. Make sure you put your questions and comments down below so I can go and see. Uh, Am I coming through? That's the next question. 'Cause I don't see. Oh, wait, there we go. That's weird. I don't know why I'm not showing up on the screen over here. But yeah, you know the deal. I'm gonna go and share this over on Facebook. That way, we are ready to rock. But yeah, so a couple of questions we're talking about tonight are herbs and dogs, and a little about seizures and fevers, and what we can do about that to avoid letting that happen. So, yeah. A little bit of a problem. I don't know what was going on. I'm gonna check that out for later to see what the hell, why my internet wouldn't let me change. It was annoying. I don't know what was going on with that, but apparently I wanted to keep talking over there. All right, so what we're gonna do is we're gonna pull up real quick. We're gonna pull up some literature on those herbs and dogs we were saying. So we're gonna start out talking specifically about turmeric, curcumin, and that was what we're talking about ideally. So effects of and synergy of feed ingredients on canine neoplastic cell proliferation. So we're talking about cancer, right? Neoplastic, new growth of cell proliferation. We're talking about overgrowth of those cells. So now, proliferation can be a beneficial growth if we're not talking about a cancer, talking about a benign situation, cell regeneration. But in this specific case, we're talking more, of course, about cancer. So that was kind of actually what the question initially started out with. We were talking about how it can be problematic in, how it can be problematic in dogs who have overgrowths. So we're going to pull up as the PDF here, though. We have the study. This was from last year, 2016. And adjunctive use of nutraceuticals in human cancer has shown promise, but little work has been done in canine neoplasia, so dogs. Previous human research has shown that polyphenols and carotenoids can target multiple pathways, either by increasing or decreasing the effectiveness of these drugs. Considering the routine and the well-controlled feeding, and oh, you know what? Sorry about that. Looks like you can't see the whole screen, can you? So let's see. We're gonna do. So we're gonna condense that in that way. There you go. Now I should be able to read that. Well-controlled feeding practice of most dogs. The use of nutraceuticals incorporated into pet food is attractive, pending proof that the extracts are able to improve mission rates. The aim of the study was to examine 5 feed ingredients for anti-proliferative effects, as well as interaction with tersarinib phosphate and doxorubicin hydrochloride when treating canine neoplastic cell lines in vitro. Screening using MTT proliferation assays showed that green tea, turmeric, and rosemary extracts were the most effective. Turmeric extract was the most potent and exhibited synergy with rosemary extract at concentrations from 1 to 25 nanograms per milliliter. This combination has an additive or synergistic effect with chemotherapeutic agents at selected concentrations within each cell. No significant effects on cell viability were observed when combination therapy was used with normal primary cells. So basically saying that there wasn't really too much being changed when we were talking about using the chemotherapy. So we're talking about using chemotherapy along with the herbal combination. They're basically saying that there was no change with normal cells, whereas with neoplastic cells, cancerous cells, there was a change. The use of turmeric and rosemary extracts in combination may be worthwhile to investigate in preclinical and clinical neoplastic, considering there are no negative effects on traditional chemotherapy treatment. Furthermore, further studies into the pharmacokinetics and mechanisms of action of these extracts should be investigated. So we're talking here again, join in here, talking about cancer and dogs, but generally speaking, herbs and different things, whether they're good to go with dogs. So right now we're talking specifically about turmeric and rosemary, showed some tremendous potential. We're going to dig through it a little bit more. So neoplasia is the cause of death in 20 to 30% of dogs in the United States. Incidence and mortality increases to 50% if dogs are over the age of 10. So unfortunately, just like humans, dogs get cancer. Not a good thing. So ways of being able to fight that could be a good thing. The incidence and in some cases tumor origin are different between dogs and humans, however, with lymphoma, mast cell disease, osteosarcoma, and mammary neoplasia being particularly prevalent. Can't talk tonight, can I? Prevalent in dogs. Current treatments rely on the use of chemotherapeutics, radiation therapy, and when practical, surgical interventions. What does that sound like? Oh yeah, same nonsense they try on humans, right? Yep, it is what it is. So the efficacy of such treatments varies greatly and relies heavily on the stage of cancer progression when treatment is provided, genetic predispositions, and tolerance to the chemotherapy and or radiation treatments. So the use of natural compounds or nutraceuticals in combination with traditional chemotherapy might provide value, and 40% of pet owners admit to using nutritional supplements as a complementary therapy. Natural products have been used in the initial design of chemotherapies, with well over half of all compounds, synthesized or screened, being more potent derivations of natural products. The effects of these compounds, whether natural or synthetically made, act through a variety of mechanisms that may contribute to the anti-cancer properties. So, basically, the synergistic effects of these compounds could be beneficial. So in this one, what they ended up doing is they tested a number of different herbs and they tried to see what they could do compared to chemotherapy and in conjunction thereof, or conjunction therewith. And look at that. They were dissolved into 100% dimethyl sulfoxide, more popularly known as DMSO. So those of you who know about the fire cream, we've talked about using that to be beneficial. Using that along with some of these herbs could be beneficial in fighting those cancer cell lines. Now, again, I do believe I said the study was done in vitro, though, so obviously it's not going to be as beneficial. Yes, these are cancer cell lines in vitro, so obviously not going to be as effective in terms of the net result effect, but still, bear with us here. So the herbs that they went and tested were black pepper, green tea, pomegranate, rosemary, and turmeric. All right. What they did, so they did them in conjunction as well or isolated, and they found that certain ones did work better together. Now let's move through. Results: single treatment versus dual combination treatment on 3 types of cancer cell lines. All 3 cancer cell lines were sensitive to turmeric extract with an IC below 13 nanograms per milliliter, and a significant decrease in cell proliferation at concentrations of at least 0.8 nanograms per milliliter in the C2 cell line and concentrations of, RE also had an effect with IC50 less than, we don't really need to go through all that. That's going to scare you all off already as it is. Low concentrations of rosemary extract cause a minor increase in proliferation when used alone, a 9% increase, an average of 11% increase for concentrations of 3.1 nanograms per milliliter, etc., etc. Green tea, black pepper, and pomegranate extracts required a concentration greater than 20 nanograms per milliliter to reduce cell proliferation. And they have a table for that. However, the most effective and consistently synergistic combination was turmeric and rosemary extracts. No other dual extract treatments were found to have considerable or consistent synergy at any combination of extract across all 3 cancer cell lines. Data not shown for -4 combinations. Combination of turmeric and rosemary resulted in a decrease in the concentrations of each extract needed to reach an IC50 in all 3 cell lines, suggesting a synergistic combination. So we'll take a look at those charts in a second. But essentially, again, turmeric and rosemary together seems to be beneficial at reducing the rate of cancer cell growth in dogs. Further increases, as we go going down, rosemary extract was reached in the C2 cell line and concentration of 12.5 nanograms per milliliter. In the turmeric plus 12.5 nanograms per milliliter of rosemary extract. And D17 cell lines. Further increases in inhibition were not detected at higher concentrations when compared to DMSO treatment alone. So DMSO treatment alone was enough to suspend some cell proliferation. That's pretty cool. Natural extracts and chemotherapy interaction on growth inhibition of cancer cell lines. The CI, or confidence interval values, generate from the C2 cell lines with the dual extract treatment in the presence of tosanarab. No, I'm not going to try pronouncing that anymore. The chemo, phosphate, showed that both extracts were added at 0.8. Again, the numbers really aren't that important for right now. Screw it. So, oh yeah, is that the first chart for turmeric? Yes. So looking at this here, we have percent proliferating cells of control, and then we have what is our key? Dual extract, so it's the rosemary plus turmeric is the straight bar line. The rosemary is the dotted line, and the turmeric is the dashed line. And so we're looking here. The amount of cells that are proliferating, depending on the dose, when we get to a dose of 25 milligrams, or sorry, nanograms per milliliter, we see precipitous drop-off versus on the first one. So rosemary's on top. Rosemary was the least effective. Turmeric was more effective, and in conjunction, we see an even better increase. The ideal dosing, looks like it's about here, about that 6.3, 12.5. You see the point where we start to see a flat lining. So past 12.5 of each probably isn't going to be that big of a deal. Probably need that much more. All right. From there, we're looking at B's percent of proliferating cells of control. Oh, let's see what A, B, and C are. Oh, different types of cancer cell lines. So there is the C2, the CMT12, and the D17 cell lines. So the different various types of cancer cell lines. If we look here, actually, interestingly, on the second type of cancer cell lines, after 12.5, we actually see an increase in proliferation again. It's absolutely minor. It looks like it ranges from like 20% to like 20 back to 25, but even so, 12.5 seems to be that magic number where we start to see everything really dropping off for all three. And then here we have the actual ratio of the turmeric dose to the rosemary extract dose. So this right here are the confidence intervals, or rather, the combination index. They indexed it together, and again, that 12.5 seems to be the magic dose here. Either way, the use of bioactive polyphenols and/or carotenoids from feed ingredients is well studied in human cancer cells, and there may be efficacy in the utilization of many of these bioactive components, particularly in combination. In the current study, we surveyed 5 extracts, which were selected based on initial screen of feed ingredients, most abundant antiproliferative compounds that could hinder cell proliferation, which would represent the high end of physiologically achieved concentration approximately. Our results indicate that turmeric extract was the most potent at inhibiting proliferation at low microgram dosages. Have I been saying nanogram? I'm shot. To further confirm the single extract and potential interaction of turmeric extract and rosemary extract in the MTT, the modest differences in terms of inhibitory effects were in comparing the trypsin blue, and it is not really that important. Long and the short, turmeric. Here we go. It's a nice little pretty graph that we can look at. DMSO is the single bar line, so that's the control of just DMSO. When we add turmeric, we see a little bit of benefit. When we add rosemary and turmeric together, we see, let's see, hold on. CDF. All right, so yeah, there's different cancer cell lines, and we're looking at this here. Hold on. I thought I had a message there. When looking at the various cancer cell lines, we see benefit, the most benefit in conjunction of the two, whereas the DMSO in and of itself can be beneficial. So the DMSO would be able to clear things through, provide sulfur that could be beneficial, but hold on. Conclusions. This study of commonly used feed ingredients showed that combination of turmeric extract and rosemary extract diminished the growth of cancer cells. The synergistic effect was observed at 10 micrograms and below, indicating a potential for physiological effects. However, in vivo pharmacokinetic and efficacy studies are needed. Although we are unsure of the bioactive molecules inducing these effects, the high concentration of curcuminoids and carnosinic acid are likely involved. The antiproliferative effects of chosen chemotherapeutics was not hindered by these extracts when they were used in concentrations in combination at concentrations of 3.1 and 6.3 micrograms across a range of chemotherapeutic concentrations. Further testing of other chemotherapies with these specific extracts is warranted to ensure no distinct antagonism is evident. Basically saying they want to make sure that there's not going to be any kind of, contraindication for the dog. But in addition, further examination of potential apoptotic or programmed cell death effects and cellular pathways affected by these extracts individually and in combination may be fruitful in determining the similarities and differences of the effects between cell lines. All right, so what I'm going to do is I'm going to go back. I'm going to slap this article in the description. I probably should have done that before so you could have read along with me, but you know the deal. Sometimes I just don't do that first because whatever. Anyway, I'm just going to slap that into the description page. And this was this morning at Long Island Strength and Performance. We were discussing whether or not it was okay to be giving the dog some herbs, particularly turmeric, because they had an overgrowth going on. But yeah, so let's try to get a little bit of some live questions going on in here, guys. But in the meantime, what we're going to do is we're going to move on a little past that. And bam, you should be able to see that down below now. Oh. All right, so in the description, we now have the herbs for dogs. Let's move past that. Let's go a little further. We had another study that I pulled up here. This here was talking about evaluation of cytotoxicity. Cytotoxicity evaluation of curcumin treatment in DH82 canine histiocytic sarcoma cell line. So basically, this one is looking at curcuma longa or turmeric and its ability to help to clear out cancer in dogs again. So we're just going to pull this one up. And I'm just going to open this up real quick and slap it down below as well. That way you guys can see what's going on along with me. Oh. All right. So, talking about the identification of targets and new drugs to fight cancer is one of the greatest challenges for biotechnology researchers, not only for humans, but also animals that are affected by the disease. It's estimated that, for example, in the 1 in every 3 to 4 dogs will develop some type of cancer during its lifetime, twice as much as that of the human being, which we saw before in the last study that, so they said about 30%, which 3 to 4 dogs, which would be, 1 in 33% or 25%, so about 30%. And they had said that if they make it past 10, that number goes up to 50% in the last study. So, DNA methylation is an important epigenetic mechanism which controls gene expression during cell proliferation and differentiation. Basically, whether or not a methyl group is getting added on controls whether the light switch of your genes is going on. So epigenetics really determines, again, whether or not the light's on or the light's off. Different factors go and change how those genes express, and methylation is a mechanism by which that occurs. So it basically is one of the key switches that turns the light on or off. So the identification of epigenetic drugs, which are let's rewind, we skipped one. Deregulation of this mechanism is one of the events which can contribute to the development of cancer. So when the ability of those cells get turned on or off, that light switch, or not cells, the ability of those genes get turned on or off can be a problem. So that epigenetic dysregulation can be something that can lead to cancer, right? So the identification of epigenetic drugs, which are molecules that can revert aberrant DNA methylation, is one of the main areas of cancer research nowadays. In this regard, natural products are a rich source of possibilities to increase the arsenal of epigenetic drugs. So the study aims to analyze the cytotoxicity, or the ability to kill the cells, of curcumin treatments in DH82 canine histiocytic sarcoma cell line, a specific type of cancer cell in dogs. So this polyphenol derived from curcuma longa has emerged as a potential multimodal cancer-preventing agent which modulates multiple cell signaling pathways and has also been described as an inhibitor of DNA methylation. The knowledge of the biological effects of these substances can pinpoint targets for the development of epigenetic drugs with a positive impact for the treatment of canine and human cancer. Nice. So the DH82 cell line demonstrated susceptibility to the treatment with curcumin, which reflects the importance of this natural product as a tool for the development of new epigenetic drugs, which could be explored to fight cancer in pet dogs. Bam! Oh, what the hell? Tell me this is just the abstract. Oh, it's a poster presentation. Oh, that's not fun. I probably should have looked more at it before I went and just slapped it on in there. Fair enough. That's my fault. Either way. Is the poster available at least? Can we see that poster? Oh man. It says the poster. Well, we'll go back to this one for a second. All right. All right. So we're looking before to see what else there was available. Apparently not crazy amounts, but we saw that last one was a 2016 article there, so. Gingival healing in dog. New paradigm. Let's see if they talk about dogs in this one, because that was specifically what we're talking about. Dog. Nope. Canine. Nope. So that one, unfortunately, is not the winner either. C'est la vie. We get the point. What we are going to move on to, though, now, we're going to look at cannabinoids and fever. So in this specific case, we're talking about viral infections. Now, the reason why it's important, why? Well, what happens oftentimes when we have some type of cold, we have a fever. Why? Well, that fever is a key component to combating your viral or other pathological infection. So, what we are going to do, let's see, we are probably about 24 minutes in, so I'm going to try and slap that in. I'll edit this afterwards in terms of the exact starting point. Yep. We're going to talk about CBD and fevers, and then CBD and seizures, because that's a key primary concern. When we have seizures, fevers that pass a certain level, we end up seeing potentially seizures, right? So epilepsy, of course, being a primary issue when it comes to seizures. Let's see. Oh, hello. Are we on that right one? What the heck? Interesting. Apparently, we have that on 2 pages right now. Just gonna slap that on there. Damn. All right. Waiting. All right. Oh. This guy's a little notification there. Awesome. All right. Moving forward. Let's go actually take a look at this study now that we slapped it on in there. We have exogenous cannabinoid receptors or receptor agonists may influence cellular and systemic host responses. The anti-inflammatory activity of the cannabinoids may compromise host inflammatory response to acute viral infections, but may be beneficial to persistent infections. In neurons where innate antiviral pro-resolution responses include the activation of NOS1, inhibition of calcium activity by cannabinoids, increased viral replication, and disease. This review examines the effects of cannabinoids and their agonists in viral infections. So let's pull up the PDF here. And actually, again, I'm just going to slap that on in because I ended up putting not the PDF, which again, I prefer PDFs. Just my own personal OCD. So that'll be down in the description. All right. Both endogenous and exogenous cannabinoids can influence the course of infections in vitro and in vivo. This review will focus on viral infections in mammals, but will also describe what is known about other infections. The biggest key here has to do, again, with their inflammatory aspect. So we're just going to fast forward more towards their roles in inflammation. When a host is infected with a virus, there's dynamic competition between the ability of the host to first marshal innate hours to days and then adaptive immunity about 7 days after the infection versus the replication and spread of the virus first within the host and then to additional susceptible individuals. When a virus is able to outpace the containment efforts, the host may succumb. Pathology may result from damage to tissues by viral-induced cellular apoptosis or necrosis, or alternatively, host immune response may result in immunopathology or the perceived symptoms of the infection. If, however, the innate and adaptive immunity successfully suppresses viral replication, specific lifelong immunity may result. So, talking here, inflammation is essential for the recruitment of both innate and adaptive immune cells to the site of infection to control virus production and limit spread, and then to promote recovery. Inflammation is comprised not only of non-specific cells. Sequentially, these are the polymorphonuclear leukocytes, natural killer cells, and macrophages, and then pathogen-specific T lymphocytes recruited from circulation and activation of antibody-secreting B lymphocytes, but also the induction, production, and secretion of cytokines, chemokines, interferons, complement components, acute phase reactants, reactive oxygen, and nitrogen intermediates, and other mediators. Yes. So, many of these pathways may be impaired or compromised when endogenous or exogenous cannabinoids are present during infection. So basically what we're saying here is that short term, in terms of dealing with an inflammation during a cold, it may not be the best idea to just crush that because you need that inflammatory response, is basically what they're saying. When it gets to the point that it can be problematic, that's when we're talking about it. The more important need for it. So people often talk about, if your temperature goes above a certain point, if you have a fever, like it's way too high, you need to immediately run to the doctor because you're going to have a seizure, right? So that's what we're talking about now. We're past the point where we're trying to use the inflammation to be beneficial mediator. We're at a point now where we need to clear that out. So, moving forward, there's various viruses and how their effects can be changed by various CBD, or rather by cannabinoid specifically, so not specifically CBD, but cannabinoids in general, what the various ones can do and how they, this is based on actual studies. So if you really want to go through that, there's quite a bit on the various ways that viruses can affect these, how endocannabinoid or how cannabinoids can affect these viruses. So where is it? I saw this earlier. Okay, the anti-inflammatory effects of cannabinoids have been found to be beneficial in attenuating fever induced by a bacterial endotoxin, inhibiting cytokine responses to the Corneobacterium parvum endotoxin. These drugs may also offer therapeutic efficacy in meningitis caused by Streptococcus pneumoniae and in irritable bowel syndrome. So, two excellent reviews of the impact of cannabinoids on bacterial, yeast, and protozoan infections were published in the same issue of the Journal of Neuroimmunology. Basically, they concluded, both reviews conclude that THC significantly reduced host resistance to infection, and similar host compromise will be found in man. In more than 12 years since those reviews, additional findings have extended the serious consequences of cannabinoids on host responses to pathogens and opportunistic infections. So, It is a risk factor for TB infections, but this author speculates that the suppression of host innate immunity by THC contributes to the increased severity of tuberculosis in users. Similarly, more serious exacerbations of central nervous system infection has been attributed to marijuana consumption, possibly by inhibiting macrophage chemotaxis. They may relieve pain and may induce hyperphagia, which could be beneficial in cancer. However, these physiological characters are not relevant to most viral, bacterial, fungal, or parasitic infections, where the regulation of inflammation is central to controlling pathogen replication and immunity. However, the same anti-inflammatory properties of cannabinoids are described as detrimental to the host in handling the other infections. In most cases, a rapid and robust inflammatory response associated with the production of pro-inflammatory cytokines and effector T lymphocytes capable of eliminating infected cells is essential to recovery and survival. So, basically, breaking that down, short term, if we are dealing with the actual disease itself, okay? If we are no longer edible, if you're dealing with a condition, whether it be a pathogenic infection, whether it be virus, bacteria, etc. Short term, you need that fever to be around to fight that infection. However, sustained temperatures that exceed high levels can result in deleterious effects. It can result in problems. As such, that would be the time when CBD can be beneficial in terms of where it's saying here that it's problematic because you need that inflammation to respond to the infection. After a certain point, inflammation becomes a problem. We talk about how stress is, the allostatic load. Up to a certain point, stress is an excellent thing. Short, acute, intermittent stress, great thing. In the case of an infection, you want to have that inflammation. However, when it gets to be sustained, that's when it gets to be problematic. So, while taking it all the time, if you have a small short-term infection, might actually be detrimental. Long-term, if you've had that cold for a while or in an acute instance of a super high fever where you need to get it down ASAP, that's where it gets beneficial. So, cannabinoids are profoundly anti-inflammatory and may impair calcium-dependent enzyme systems, which are central to inflammatory and cell-autonomous antiviral responses. When viral-induced host responses lead to immunopathology, as seen in the rodent model of multiple sclerosis, etc., or in a persistent infection of the central nervous system caused by non-lytic virus, can't say letters, right? BDV, cannabinoid treatment was beneficial. In all other viral infections, both in vitro and in vivo, it led to disease progression, increased pathology, and sometimes even post-death. Therefore, in many clinical settings, including latent infections caused by HIV or HSV1 and persistent infection of the liver caused by HCV, they have worsened disease outcome. Now, to be clear, this particular study was talking about cannabinoids as a total class, and if you look back to it, if we look here, THC, marijuana, cigarette, THC again. A lot of them are exploring THC, oral cannabinoids. We were talking more anandamide, which is a synthetic. A lot of these are THC related. That being said, the reduction in inflammation can overall be a beneficial thing. So we have here a different one, cannabinoids in epilepsy. And again, epilepsy, of course, being the seizure-based issue. Not the, but, one of the. Basically, that it's been used for centuries to treat seizures. Recent anecdotal reports, accumulating animal model, and mechanistic insights have raised interest in cannabis-based anti-epileptic therapies. In this study, we review current understanding of the endocannabinoid system, characterize the pro- and anti-compulsive effects of cannabinoids, such as THC and CBD, and highlight the scientific evidence for preclinical and clinical trials on cannabinoids in epilepsy. These studies suggest that CBD avoids the psychoactive effects of the endocannabinoid system to provide a well-tolerated, promising therapeutic for the treatment of seizures, while whole plant cannabis can both contribute to and reduce seizures. While we discussed the results from a new multi-center open-label study using CBD in a population with treatment-resistant epilepsy. In all, we seek to evaluate our current understanding of cannabinoids in epilepsy and guide future basic science and clinical studies. Hey, Megan, what's up? We have one of our official Fire Cream athletes, which again, make sure we're checking that out later. Sorry about that again towards the end, as always, because we are hopefully launching soon. I'm assuming you've been lurking around for a little while, but hey, what's up? So yes, I'm going to slap the last one here for now into the description. You can go check this study out as we're going through it. Apparently I have that twice there. And we'll just say that's about like 35 minutes. Sure, why not? So if you go down in the description, you'll be able to see the study right here. We're going to pull it back up. I never really put it away for you guys. You guys saw it the whole time. So it's been used for centuries to treat seizures, as we said. Epilepsy affects 2.9 million people in the U.S. and 65 million people worldwide. It's a crazy statistic. But 1 in 26 people in the USA will develop epilepsy in their lifetime. Characterized by recurrent seizures, epilepsy encompasses multiple disorders caused by various etiologies, including genetic syndrome, stroke, infection, and traumatic brain injury. So that's what we're talking about right there before infection and fevers, and we're doing that there. Don't have a topic today, but I like the cannabinoids topic. Awesome. Yeah, we have, again, I know people that have had seizures, and I know people that have had super high fevers that are afraid of moving towards a seizure, and as such, keeping it rolling. All right, so many patients with epilepsy also have sensoromotor, cognitive, psychological, psychiatric, and social impairments, as well as impaired quality of life and increased risk of premature death. While epilepsy can affect patients of all ages, it most commonly affects children, the elderly, individuals with low socioeconomic status. That's social justice, guys. The estimated direct and indirect annual cost of epilepsy in the United States is $15.5 billion. So while many drugs can limit seizures, no drug can prevent the underlying cause of epilepsy or the development of epilepsy in patients who are at risk after head trauma. A third of patients remain pharmacoresistant, failing to achieve sustained seizure freedom after 2 or more adequately chosen. So this is getting more into the in-depths of epilepsy. We've talked before about the benefits of fasting and or ketogenic diet for epilepsy. We're not focusing so much on that for right now. What we really want to look at more is cannabis, or specifically CBD and epilepsy. But apparently it's been used for millennia for medical, recreation, and manufacturing purposes. Around 2900 BC. I'm not adding the E, screw off with that. The Chinese Emperor Fu Sui, Fuxi, characterized cannabis as having sacred yin, weak passive forces, and yang, strong active forces. Features, suggesting that it can restore homeostasis to an unbalanced body. And today, that's actually kind of been followed up on. We know that CBD acts as an adaptogen, meaning it helps to restore, or rather, help your body adapt. It doesn't really help. It doesn't really suppress or boost. It helps to modulate, helps to regulate. So yeah, that yin and yang aspect, another way of skinning the cat, right? So physicians in ancient Egypt, India, Persia, Rome, Arabia, and Greece used cannabis for spiritual and medicinal purposes, including menstrual fatigue, gout, rheumatism, malaria, beriberi, constipation, pain, and absent-mindedness. Wait, you use cannabis for absent-mindedness? Anyway, so early documented use of cannabis to treat seizures includes a Sumerian text from 2900 BC and Arabian document from the 12th century. That is a ginormous jump. So in terms of the early documented use, 3,000, so 5,000 years ago, and then 800 years ago, which, I mean, that's cool. 800 years ago is still a long, long time ago, but it's kind of funny how it's like, and jump. So since 1854, the U.S. dispensary listed cannabis to treat neuralgia, pain, depression, muscle spasms, insomnia, tetanus, chorea, one of those two, insanity, and other disorders. It was valued for its analgesic, anti-inflammatory, appetite-stimulating, and antibiotic properties. So in the mid-1800s, the British surgeon William O'Shaughnessy reported cannabis therapy as a treatment for epilepsy, recounting an alleviation of pain in most and remarkable increase in appetite in all, unequivocal aphrodisia, and great mental cheerfulness. So, yeah, sounds about right. J.R. Reynolds and W. Gowers also noted the benefits of cannabis epilepsy. Gowers reported a man who previously failed bromides, whose seizures were controlled on 9.8 grams of cannabis indica dosed 3 times daily for up to 6 months. That's a lot of cannabis. Anyway, was first regulated in 1906. The follow-up 1937 Marijuana Tax Act was opposed by the American Medical Association, which considered the more severe restrictions an infringement on the physician's freedom to treat patients. Then 1970, Clinton changed even more, moving past. So let's move past. Let's get to the point where we can start to see where CBD can be beneficial. So it's THC. We have phytocannabinoids, classification and function. CBD resembles THC structurally, but the two molecules differ significantly in their pharmacology and function. As you see here, we have precursors, cannabigerolic acid, which then can go towards THC or CBD. And as a small side note, people often talk about, well, will I test positive if I start to take CBD? And the fact of the matter is, it is not the same pathway, so it's going a different route. But anyway, potential targets for CBD are reviewed in detail in another article, so yeah, look at that one if you're so inclined. But it exerts dynamic control over intracellular calcium stores through multiple activity-dependent pathways. Induces bidirectional change in intracellular calcium levels that depend on cellular excitability. Again, beyond the scale we're trying to cover for right now. Antagonizes GPR55, which functions as counterpart to the canonical CB1R and CB2R receptor signaling pathway. Yeah. So it's the CB receptors, which are your cannabinoid endocannabinoid system. So the CB1R and CB2R signaling pathway. Okay. It regulates several transporters, enzymes, and metabolic pathways that are in common to delta-9, THC, and endocannabinoid system, but inhibits the uptake of adenosine by blocking the equilibrative nucleoside transporter. So, CBD also inhibits the uptake and enzymatic degradation of anandamide. So that's the synthetic one. If you were to take CBD with anandamide, which is the synthetic version of it, of THC, it basically slows down the breakdown of that, making it last for longer. So it elevates anandamide extracellular concentrations. So they are like so. Thus, dynamic interactions likely occur between multiple cannabinoids, such as CBD and THC itself, which leads us to the entourage effect. Basically, the different cannabinoids work in conjunction to reduce those issues. So CBD limits inflammation and oxidative stress. CBD reduces oxidative toxicity in vitro glutamate excitotoxicity, say. Basically, in a cell line, glutamate, people think about MSG, it is something that causes overexcitation, which is toxic to the brain, so excitotoxicity. And so by taking the CBD, it reduces that stress, it reduces the oxidative toxicity. It raises adenosine to oppose lipopolysaccharide and induce inflammation and tumor necrosis factor alpha release. So the LPS is found in the cell walls of pathogenic bacteria, in the microbiome. Tumor necrosis factor alpha, again, is one of the initial chemical mediators we release during the inflammation process. Yeah. I got an itch I can't get rid of. Nice. So neuroprotective action may result from reduced myeloperoxidase activity, neutrophil neutrophil migration, and microglia high mobility group box 1 expression. All right. Regulates the inflammatory response to neuropathic pain. Taken together, these studies suggest CBD reduces neuroinflammation in several disease-specific conditions. So, moving past that. Preclinical evidence of cannabinoids in epilepsy. Multiple animal models demonstrate the efficacy of cannabinoids in preventing seizures and reducing mortality in epilepsy. Animal models highlight dynamic changes in the endocannabinoid system following chronic seizures, with both acute and chronic homeostatic regulatory components. So again, we have some studies here that are being basically summarized and how they are being affected. Basically, type of seizure model, the method, the mechanism, relevant human condition, and the common use. All right, as I look ridiculous, glancing sideways. So phytocannabinoids, evidence from 34 studies from 6 animal species demonstrate that THC is anticonvulsant in 61%, proconvulsant in 3%, mixed in 3%, and shows no significant different effect in 32.4%. Even though I went and said 3% for 2.9. Whatever. Anyway, so potentiated effects of phenytoin and phenobarbital, which are common anti-seizure medications, in the maximal electroshock model of generalized seizures. So basically here we're looking at the relative expression, and here homeostatic change to hippocampal cannabinoid type 1 receptors, so CB1R, in preclinical animal seizure models. GABA, which is gamma aminobutyric acid, is right there. And we're looking here the normalized expression in healthy tissue and how those are affected. Okay. But anyway, they noted a proconvulsant effect of THC in rats and mice, although species-specific differences in the reception, in the endocannabinoid receptor expression, may be what changes up the responses to THC, CBD, and its homologue, cannabidiol. Cannabidiol. Yeah, that one. Were 80% anticonvulsive and 19.5% ineffective at reducing seizures in mice and rats. Notably, no studies showed a proconvulsive effect for CBD or CBDV. Okay, it potentiated the effects of phenobarbital, ethosuximide, and valproate in 2 seizure models. So these studies suggest that both delta 9, THC, and CBD provide significant protection from seizures in preclinical trials, presenting potential targets for human studies. Now we get to our clinical evidence. All right. So, in terms of clinical evidence here, let's actually zoom in on that a little bit. Cannabinoids and preclinical seizure models. I'm talking about animals here. So there's a nice little graph of what we were just talking about before. Short-term answer, basically things that can regulate the endocannabinoid system as a whole generally have an anticonvulsive effect, a mixed effect, or no significant effect. So the majority of them are anti-inflammatory, anticonvulsive. If we are talking about the CB1 receptors, again, ones that are agonists, ones that act on them positively, well, again, it's a very high anticonvulsive effect. In fact, we are looking at here, roughly 70% are anticonvulsive. If we are looking at CB1 antagonists, so ones that have a deleterious effect on the CB1 receptors, those will generally have no effect. They have for no significant effect. We're looking at 55.6%, whereas we do have a moderate proconvulsive effect of about 40% there, so 38.9. If we're looking at THC, relatively very anti-convulsive with a rate of 61%, and again moderately high no significant effect. So very minor proconvulsive. It could occur in 2.9%, whereas in the no significant effect, meaning nothing really happens, at 30%. Then we get to the CBD and its homologue, CBDV, and we have super high anticonvulsant effect, 80.5% throughout two species in the studies, with a no effect in 19.5%, so basically nothing. Now, to be fair, the number of discrete conditions and miles, we're looking at 41. This is a very low sample size in this particular study. That being said, both from some of the clinical and the empirical observational effects I've seen in people and personally worked with the clients and stuff, definitely seen some tremendous benefit from it overall. Anyway, moving forward, clinical trials. A recent Cochrane review assessed 4 primary clinical trials to examine the efficacy of medical marijuana in seizure control, summarized in Table 2, adapted from 11 and 13. 2 of those studies demonstrated a partially anti-seizure effect of CBD, while 2 showed no significant effect. However, all 4 studies included significant limitations, including low study sizes, insufficient blinding or randomization, or incomplete data sets. The authors of the Cochrane Review and recent meta-analysis both emphasized the need for follow-up placebo-controlled, blinded, randomized controlled trials examining the role of CBD in seizure control. So that's the biggest problem. Because of those laws that we talked about briefly before I rushed past them in the beginning, from the 30s, from the 70s, and so on, a lot of the studies are just starting. And because of that, the sample size has been small, a lot of it have been in animals and whatnot. So preliminary preclinical and clinical evidence reveal the therapeutic potential of CBD to reduce seizures with high tolerability and low toxicity. And we talked about last week how toxicity was again shown about 2 months ago in a study, basically non-existent. The adverse effects pretty much not there. So accordingly, CBD represents a highly desirable treatment alternative to patients with early onset severe epilepsy, such as DS and LGS, in addition to pharmacoresistant seizures. These patients suffer from severe neurodevelopmental delay, intellectual disability, autism, motor impairments, and significant morbidity and mortality. So, basically, there was an open label study at 10 epilepsy centers, collected data on 213 patients with treatment-resistant epilepsy. That's a pretty awesome one. This predominantly pediatric population had a mean age of 10.8 years. It was added to their existing anti-epilepsy drugs. There was an average of 3 concomitant anti-epilepsy drugs. So basically, these particular kids were predominantly kids. The ages range from 2 to 26. Had an average of 3 other concurrent anti-epilepsy drugs. The average baseline was 60 per month for total seizures and 30 per month for convulsive seizures. That's a lot. The primary goal of the study was to assess the safety, but seizures diaries were obtained for convulsive drop, and total seizures to provide a potential signal regarding efficacy. 12-week or longer continuous exposure data were obtained for 137 patients and used for efficacy measures. The most common epilepsy etiologies were DS and LGS syndromes, along with Aicardi syndrome, Dew syndrome, whatever. At week 12, tuberous total convulsive and non-convulsive seizures showed a median percent reduction from the baseline of 54%, and total convulsive seizures showed a median percent reduction from baseline of 51%. In DS, CBD reduced convulsive seizure frequency by 53%, and 16% of DS reached complete convulsive seizure freedom. So 16%. So not a crazy one, but 16% still reached complete seizure freedom by week 12. Atonic seizure frequency among patients with LGS was reduced by median of 52% at week 12. AEs 10% greater than 10%. Sorry. Adverse effects. Anti-epilepsy. Are we talking adverse effects or anti-epilepsy? Probably AED is probably what they're trying to say. Anti-epilepsy drugs, including somnolent. Oh, no, duh. I should have kept reading, and that could have figured out the context of what the AE was in that case. Yes, AEs was, in fact, adverse effects, like I said at first. Included somnolence, diarrhea, fatigue, and decreased appetite. 9 patients were discontinued for those 9 patients, or 4% of the population, were discontinued for those adverse effects. The investigators concluded that CBD reduced seizure frequency across multiple drug-resistant epilepsy syndromes and seizure types and was generally well tolerated in this open-label study. Randomized controlled trials are now ongoing for DS and LGS. All right. So, going past, cannabidiol formulations, pharmacokinetics, pharmacodynamics, and drug-drug interactions. So there are 3 pharmaceutical products that are currently in trials for development. Oh boy, so once that crap goes through, we're going to be in trouble. But there is a CBD, 99% CBD derived from cannabis sativa plants in strawberry-flavored sesame oil, a synthetic CBD, of course, that was coming down the line, and transdermal CBD gel, which, by the way, Fire Cream, gonna be transdermal CBD, but other CBD-containing products are available commercially and obtained online. Yes, they are. However, the quality control and consistency of these products may vary considerably. Again, when we're talking specifically about Alchemist Closet, I can get you the COAs. It's the real deal. The quality control is second to none. Despite not having the big pharma backing, we do have pharmaceutical controlled labs making sure they are on point. Indeed, a recent study by the US FDA, tested 18 products claimed to contain CBD made by 6 companies. Of these, 8 contained no CBD, 9 contained less than 1% CBD, and 1 contained 2.6% CBD. And again, if there's any questions regarding Alchemist Closet, I can get you the COAs. So, they're real. Anyway, they are prominently metabolized within the liver, inhibit CYP450 enzyme metabolic activity, particularly the CYP2C isozymes and low concentrations. Basically, they go and change the way liver enzymes are working. Anyway. More studies evaluating the effects of CBD. So this here is basically part of their little review. You can go to the results and pull through these different ones and how the effects are there. Now, there is the magnitude of potential placebo effect in response to the power of belief. Given the social and mainstream media attention selectively reporting dramatic benefits of artisanal cannabis preparations for children with epilepsy, there are high expectations on the part of parents. The potent role of the placebo response was suggested by a recent survey of parents whose children with epilepsy were cared for at the Colorado Children's Hospital. A beneficial response of greater than 50% seizure reduction was reported 3 times more often by parents who moved to the state compared with those who were longtime residents. So they're saying that the placebo could be in effect. No differences in epilepsy syndrome, type of artisanal preparation, or other factor could account for this difference. While studies have reported a significant placebo response in adult patients, such as those with Parkinson's, placebo response rates are particularly high among children and adolescents in subsets of disorders, including psychiatric, anxiety, depression, and OCD and ADD, medical asthma, and painful migration gastrointestinal conditions. As the current RCTs, or randomized controlled trials of CBD, primarily target children with severe epilepsy, this may be an important issue. Among parents with treatment-resistant focal epilepsy, a meta-analysis found that the placebo response in children was significantly higher than in adults. While the response to the anti-epilepsy drug was not significantly different in children and adults. In one predominantly pediatric LGS trial, seizures were reduced in 63% of placebo-controlled patients and 75% of drug-treated patients. Paradoxically, the intense interest and strong beliefs in the efficacy of cannabis for epilepsy may elevate placebo responses and make it more difficult to demonstrate a true benefit. So that does get into a little bit of a squirrely neighborhood. Placebo always can be a problem overall. But the thing with that is you would think that in children you wouldn't see that a placebo response to that level, mostly because in adults, we're the ones who are generally told, or rather are seeing it in social media, and seeing the results and wanting to believe that and would have that power of belief. But I'm still leaning towards do the CBD. The problem, of course, is that DEA does classify cannabis and products derived from cannabis plants as Schedule 1 drugs. Ugh. However, as they go on to point, they are the most dangerous drugs and have no currently accepted medical use, with potentially severe psychological and physical dependence. It is thus paradoxical that opiates and benzodiazepines, which have a much greater potential for psychological and physical dependence than cannabis, are classified as Schedule II drugs. With regards to the DEA's claim that cannabis-derived drugs currently have no accepted medical use, therapies such as nabiximols and other products have been approved by regulatory agencies in over 20 countries. These approvals are based on randomized controlled trials that establish efficacy and a favorable safety profile, including low potential for abuse. The Schedule I characterization makes it challenging for investigators to study cannabis-derived cannabinoids in basic and clinical science. There's often a long and costly process to secure approvals, inspections to obtain cannabinoids, to purchase a large safe, the weight of which may require clearance from engineers, and add security systems to the room and building in which they are stored. Schedule I designation often prevents patients who live in developmental centers or residential homes from participating in clinical trials. The threshold of effort for basic and clinical investigators to study cannabinoids remains as high as ever, while the availability of these substances for parents is expanding rapidly. This has created a widening gap between knowledge and exposure, as especially relevant concern in children for whom safety data are largely lacking. Again, I completely agree there should be some more research being done into that potential. That being said, the fact that the law is as it is currently, it is creating some major issues. In order to have these studies be done, we are going to need to change that law and make it not so crazy. And actually, I'm going to slap into here, up in the top corner, maybe that corner. We're going to add a card. Let's see, my fingers go up. We're going to add a video about why that could be the case. Why various herbs and are treated like controlled substances. Okay, so basically things like CBD, things like ephedra, things like kratom, why are they treated as if they are the most deleterious things you could possibly consume, yet, like I said before. Yeah. Yes, so that's that corner up there. So, yeah. Anyway, yeah, so you can drop in that corner over there. You'll be able to see now, basically, why that is. You can go check that out. It's another video over there. But, basically, finishing up with that thought, in conclusion, for over a millennia, preclinical and clinical evidence have shown that cannabinoids to CBD can be useful to reduce seizures effectively, particularly in patients with treatment-resistant epilepsy. However, many questions still remain. See Box 1 regarding the mechanism, safety, and efficacy of cannabinoids in short and long-term use. Future basic science research and planned multi-center placebo-controlled clinical trials will provide insight into cannabinoid function and potential neuroprotective effects of the endocannabinoid system. These findings will increase our mechanistic understanding of seizures and may provide novel, targeted therapeutics for epilepsy. Short version, CBD for seizures, good. If we are talking about a super high fever, and we're talking about that in the case of dealing with an infection, again, good idea. Other good ideas? The fire cream. We've talked about that a bunch of times as well. So fire cream is great for bringing that down. As paradoxical as that may seem. However, the secondary product we're releasing without the cayenne may be beneficial as well, but stay tuned for that. Short version, basically the reduction in inflammation should be able to help to clear up that issue. Just circling back for a second, remember, we were talking before about the effects of turmeric and cancer on dogs, so different herbs, particularly turmeric, good idea for your dogs. Make sure you get ready to check out the fire cream, and I will, pull it out real quick. Bam. So if you haven't already headed on over, make sure you head on over and go like the fire cream. Go get it. Oh, Stephanie messaging me, making the cut. Hey, reached 200 likes. Awesome. So, hey, and then Megan, who is on the chat right now, our newest Fire Cream athlete. So, everybody, it's up to Megan making the cut. So, again, we are trying to get the second product out there as well. We will be launching the Fire Cream officially super soon, and don't need to show you the analytics there, but make sure you head over to facebook.com/thefirecream. It's down in the description. You can go do that. Like and subscribe, share it. I definitely want to get back to doing some daily videos, at the very least, moderately daily videos. Right now, things have been a little too crazy with the launch and with school and stuff. So, staying with the live streams. We are definitely still staying with these live streams. Would love to have some more engagement like we used to have, but I'm fine just, rambling on for a little bit and, saying hi to people as they come along. Either way, guys, if you're watching on the replay, love if you're sticking around this long. Megan and whoever else has been lurking all night, because I've seen there are a couple people here who haven't been commenting. Whoever's here, guys, thanks again. Love you. And if I don't see you tomorrow, probably won't. I'll see you next week. I'm going to stop this eventually. And